Today, we're diving into one of the most diverse and misunderstood groups of organisms in biology, the protists. This group includes everything from microscopic plankton that fuel ocean webs to parasites responsible for major human diseases and even giant seaweed you might recognize along the coasts. Protists don't fit neatly into a category of plants, animals, or fungi, and that's exactly why they're also very important. As we move through this chapter, we'll focus on how protists are classified, how they function at the cellular level, and why they play such a critical role in ecosystems, climate regulation, and the human history. By the end, you should see protists not as the afterthought, but as a central piece of the story of eukaryotic life. Protists are often introduced as the group defined more by what they are not than what they are. They are eukaryotes, meaning their cells contain a nucleus and membrane bound organelles, but they don't fit neatly into the plant's animal or fungal kingdoms. Most protists are single celled, although there are some that have large and more complex forms. You'll find them in almost everywhere water exists, from the oceans to lakes and soils, to even inside other organisms. Their diversity makes protists one of the most fascinating and challenging groups to study. Protists play critical roles in ecosystems and human health. Many photosynthetic protists, especially algae, are primary producers that support aquatic food webs and contribute to significant portions of the world's oxygen. Other protists feed on bacteria and organic matter, helping recycle nutrients. At the same time, some protists are parasites that cause serious diseases in humans and other organisms. One of the key ideas of this chapter is that protists are not simple or a uniform group. They vary enormously in how they move, how they obtain energy, and how they reproduce. Some behave like plants, some like animals, and others switch strategies depending on conditions. Because of this diversity, protists give us important clues about the early evolution of eukaryotic cells and how complex life diversified over time. Life on Earth is broadly divided into three domains: archaea, bacteria, and eukarya. The first two domains include all prokaryotes, organisms who lack a nucleus, while the domain eukarya includes all organisms with complex membrane bound organelles. Because early life forms were microscopic and some soft bodied, the fossil records gives us only limited clues about the earliest members of these groups. As a result, many of these steps that led to the last common ancestor of modern eukaryotes remains unknown. To fill in those gaps, scientists rely on comparisons among living organisms alongside the new fossil records that exists. The earliest fossils have belonged to bacteria, most likely cyanobacteria that date back over 3.5 billion years. These organisms had relatively large and complex cells compared to other prokaryotes, which made them more likely to fossilize. Most prokaryotic cells are extremely small, only 1 or 2 micrometers, and are rarely preserved in rocks. Fossilized stromatolites show that some early prokaryotes lived in these structures, cooperative communities. Interestingly, a similar interaction between ancestral cells may have played a role in the origin of prokaryotes and eukaryotes. Fossils consistent with early eukaryotic cells, which are much larger, begin to appear in the geological record about 2.1 billion years ago. When biologists compare fossils with the genomes of living organisms, a clear pattern emerges. All modern eukaryotes share a single common ancestor. Even though today's eukaryotes are incredibly diverse, most protists to plants and animals, they retain core features that trace back to their ancestral cells. Some of these traits only appear during certain life stages or in certain groups, but their widespread presence tells us that they must have existed early on. What truly defines eukaryotes is the presence of a nucleus enclosed by a nuclear envelope. Beyond that, eukaryotic cells share a suite of features that support complexity, including mitochondria, a dynamic cytoskeleton, a linear chromosome organized in histones. All eukaryotes divide their nuclei by mitosis, and sexual reproduction involves meiosis, is unique to this domain. Traits like flagella, cilia, and even cell walls may be absent in some modern groups, but their distribution across lineages indicates they were present in their last common ancestor and later lost or modified. Together, all these characteristics distinguish eukaryotes from prokaryotes and explains their remarkable diversity. Many of the metabolic processes that support life today originated in prokaryotes. Some of these, like nitrogen fixation, never made their way to eukaryotic cells. Aerobic respiration, however, is universal among eukaryotes and is carried out in mitochondria. Early in Earth's history, oxygen was absent from the atmosphere, so life depended on anaerobic pathways and fermentation. Eventually, some prokaryotes evolved photosynthesis using sunlight to convert carbon dioxide into organic molecules. Cyanobacteria played a crucial role by using water in photosynthesis and releasing oxygen as a waste product. At first, oxygen was dangerous, damaging many biological molecules. This selective pressure led to the evolution of protective pathways, including aerobic respiration, which also produces far more ATP than anaerobic processes. Aerobic metabolism became widespread among certain prokaryotes, especially alpha proteobacteria. As oxygen accumulated globally around 2 billion years ago, eukaryotes appeared in the fossil record. This timing supports the idea that rising oxygen levels and mitochondrial metabolisms were tightly linked to eukaryotic evolution. As cell biology advanced in the 20th century, scientists recognized that mitochondria are responsible for aerobic respiration, producing ATP using oxygen as the final electron acceptor. In the 1960s, Lynn Margulies proposed the endosymbiotic theory, suggesting that eukaryotic cells arose when one cell engulfed another that was capable of aerobic metabolism. Instead of being digested, the engulfed cell survived, and the two entered a long term, mutually beneficial relationship. Over time, they became so integrated that they functioned as a single organism. Although this idea was initially resisted, strong microbial and genetic evidence now supports it. Interestingly, while mitochondrial genes resemble those of bacteria, nuclear genes are more closely related to archaea, leaving an open important question about how those events occurred. One of the clearest distinctions between prokaryotic and eukaryotic cells is the presence of the mitochondria. Nearly all eukaryotic cells maintain them, although the number varies widely depending on how much energy a cell needs. In humans, like those in the liver and the skeletal muscles, can contain thousands of mitochondria. Those organelles are relatively large and dynamic in shape, sometimes appearing rounded, elongated, or evenly branched. Even though mitochondria likely originated as free living organisms, they are now fully dependent on the host cell and can no longer survive independently. Mitochondria show multiple features that strongly link them to alpha proteobacteria. They contain their own circular DNA and use ribosomes and transfer RNAs that closely resemble those of prokaryotes. Some even use genetic codes that differ slightly from the universal code. Over evolutionary time, many mitochondrial genes were transferred to the host nucleus. While scientists compare these genes, those closely match bacterial genes, particularly from alpha proteobacteria. This gene transfer helps explain why mitochondria can no longer live independently and require coordination with the host genome. The physical structure of mitochondria provides some of the strongest evidence for endosymbiosis. They are surrounded by two membranes, exactly what we would expect if one cell engulfed another. The inner membrane is bacterial in nature and forms extensive folds called cristae that increases surface area for respiration. Mitochondria divide independently by a process similar to bacterial binary fission and arise only from existing mitochondria, never from scratch. While most eukaryotes retain mitochondria, some anaerobic lineages have reduced versions, and few represent extreme cases of mitochondrial reduction. Some eukaryotic lineages gain the ability to photosynthesize, and they do so using specialized organelles called plastids. When plastids perform photosynthesis, they are known as chloroplasts. Chloroplasts contain chlorophyll A along with pigments that help absorb light across different wavelengths. Structurally, chloroplasts are two membranes. Like mitochondria, plastids are believed to have arisen through endosymbiosis. In this case, an ancestral eukaryotic cell engulfed a cyanobacterium capable of photosynthesis. This event, known as the primary endosymbiosis, gives rise to plastids surrounded by two membranes. The strongest evidence comes from similarities between chloroplasts and cyanobacteria, including thylakoid membranes and photosynthetic pigments. Nearly all photosynthetic eukaryotes trace back to a single primary endosymbiotic event in the ancestral Archaeoplastida, although a second, much rarer event occurred at the amoeboid lineage Paullinia. Protists are essentially a convenience term. It includes all eukaryotic organisms that do not fit in the animal, plant, or fungal kingdoms. Because of this, protists are not defined by a single shared set of characteristics, and very few traits apply to all of them. Scientists have already described more than 100,000 protist species, but the number is almost certainly an underestimate. Many protists live in close association with other organisms as commensal or parasites, and these relationships are often highly specific. As a result, protist diversity is likely tied closely to diversity of other hosts, making this one of the most varied groups of organisms on Earth. Protist cells push the limits of what the single cell can do. Even though plants, animals, and fungal achieve complexity through multicellularity and specialized tissue, many protists reach comparable complexities while remaining unicellular. They do this by differentiating structures and functions within a single cell. Some protists form loose colonies that behave partly like groups of individual cells or partly like multicellular organisms. Others consist of enormous multinucleated cells, sometimes with nuclei that differ in size and function. Protists display an impressive range of metabolic strategies. Some rely on oxygen for metabolism, while others live in anaerobic environments. Photosynthetic protists are photoautotrophs and use chloroplasts to capture light energy and produce organic molecules. Other protists are heterotrophs, meaning they must obtain nutrients by consuming organic material, including other organisms. Some protists are especially flexible and function as mixotrophs, switching between photosynthesis and heterotrophy depending on whether light or organic nutrients are available. Many heterotrophic protists obtain food through phagocytosis. During this process, the cell membrane surrounds and engulfs a food particle, forming a vesicle called a food vacuole or phagosome. This phagosome then fuses with lysosomes that contain digestive enzymes, creating a phagolysosome. The enzyme breaks down food into small molecules that diffuses into the cytoplasm and fuels cellular metabolism. Any undigestible material is eventually expelled from the cell by exocytosis. In contrast, saprophobic protists do not engulf food, but instead absorb dissolved nutrients from dead organisms or organic wastes. Unlike plants and fungi, the majority of protists are capable of movement. Motility is a major advantage for these organisms, allowing them to seek out food, avoid unfavorable conditions, and respond to changes in the environment. Protists have evolved several distinct strategies for locomotion, and these differences are often tied closely to their ecology and environmental history. The three main mechanisms we see using flagella, cilia, or pseudopodia, each of which relies on cytoskeleton and coordinated cellular processes. Flagella are long whip-like structures that push or pull the cell through the environment. Cilia are much shorter but occur in large numbers and be in coordinated patterns to produce smooth, controlled movement. Pseudopodia are temporary extensions of the cytoplasm that allow the cell to anchor to a surface and pull itself forward. Beyond basic movement, many protists can perform taxis, meaning they move or towards or away from a specific stimuli. A common example is phototastics, where protists use light sensing structures to move towards light sources and support photosynthesis. Protists use a wide range of reproductive strategies, with asexual reproduction being the most common. Many reproduce by binary fission, where a single cell divides into two daughter cells. The orientation of division can vary, such as transverse or longitudinal fission, which you can see in organisms like Paramecium. Some protists, including true slime molds, undergo multiple fission and divide into many daughter cells all at once. Others reproduce by budding, where small offspring form and later grow out to full size. Sexual reproduction is also a widespread among protists, and many species can switch from asexual to sexual reproduction when the environmental conditions become stressful. Nutrient depletion or changes in temperature often trigger this shift. Sexual reproduction allows for genetic recombination, increasing the chances that some offspring will survive new conditions. In many cases, sexual reproduction leads to the formation of cysts, which are protective dormant stages and almost no metabolism. Protist life cycles vary dramatically from simple single host cycles to complex parasitic cycles requiring multiple hosts. Some protists can even alternate between haploid and diploid multicellular stages similar to plants. Protists are almost always associated with water or moisture. Most live in freshwater or marine environments, but many also thrive in damp soils and even in extreme habitats like snow. Beyond free living forms, a significant number of protists are parasitic and infect plants or animals, often with very specific host relationships. Other protists play an important ecological role by living on dead organisms or organic wastes, where they help break down material and recycle nutrients. Modern classification no longer places all protists into a single group. Instead, biologists divide the entire domain eukarya into six large evolutionary lineages called supergroups. These supergroups include all eukaryotes, not just protists, but also animals, plants, and fungi. Each supergroup is considered monophyletic, meaning all members descend from a single common ancestor. Because of this, all organisms with a supergroup are more closely related to each other than the organism outside of that group. Molecular evidence strongly supports the idea that all members of Archaeplastida share a common origin rooted in primary endosymbiosis. In this event, a heterotrophic protist engulfed a cyanobacteria that eventually became a chloroplast. This lineage includes red algae, green algae, and land plants, making it especially important evolutionarily. In fact, land plants evolved from within the green algae, whose closest living relatives are still aquatic. Members of Archaeplastida show remarkable diversity ranging from single-celled organisms to complex multicellular forms. One of the most important biological patterns seen in some Archaeplastida algae is the alternation of generations. In this life cycle, both the haploid and diploid stages are multicellular. The diploid sporophyte produces haploid spores through meiosis. These spores grow into haploid gametophytes, which then produce gametes by mitosis. When gametes fuse, they form a diploid zygote that develop into a new sporophyte. In some species, the gametophyte and sporophyte look very different, while in others they are nearly indistinguishable. Glaucophytes are not very diverse or abundant today, but they are extremely important in an evolutionary perspective. What makes them unique is that their chloroplasts still retain remnants of peptidoglycan, which is a key component of bacterial cell walls. This feature directly links their plastids to cyanobacteria, an ancestral endosymbiont that gave rise to chloroplasts. Most other archaeplastida have lost their bacterial layer, so glycophytes provide a rare glimpse into early plastids may have looked like shortly after endosymbiosis occurred. Red algae, also known as rhodophytes, are a distinctive group within Archaeplastida. Unlike many other protists, they completely lack flagella at their life stages. Most are multicellular and include many organisms we casually refer to as seaweed, though some species are microscopic. Their cell walls are especially important to biology because they contain carbohydrates used to make agar and agarose, which are essential in microbiology and molecular biology labs. The characteristic red pigment comes from phycoerythrin pigments, which allow these algae to absorb light efficiently at great depths, sometimes over 200 meters. Red algae also have a very unusual life cycle with two sporophyte stages, making them an excellent example of how diverse eukaryotic reproduction can be. Green algae are the most abundant group of algae and are especially important because of their close evolutionary relationship with land plants. Both green algae and plants share similar chloroplast structures and store carbohydrates directly in the plastids rather than in the cytoplasm. These shared traits strongly support the idea that land plants evolved from within green algae. Modern green algae is divided into two major groups, the chlorophytes and the charophytes, which differ in habitat structure and evolutionary significance. Charophytes are especially important because they are the closest living relative to land plants. They resemble plants not only in structure but also in aspects of their reproductive strategies. Charophytes are commonly found in freshwater in most environments, and their presence is often used as an indicator of good environmental quality. A familiar example is Spirogyra, which forms filamentous mats in ponds and streams. Studying charophytes helps us understand the evolutionary transition from aquatic algae to terrestrial plants. Chlorophytes display remarkable structural diversity. Some are simple unicellular organisms like Chlamydomonas, which uses flagella and other eye spots to move towards lights. Others form colonies such as volvox, where thousands of cells move together in a coordinated way and only a few specialize in reproduction. Chlorophytes also include the true multicellular form like sea lettuce, as well as extreme examples such as Chloroptera, which grows as massive single cell containing many nuclei. Amoebozoas are defined less by where they live and more by how they move. Like Archaeplastida, this group includes single-celled organisms, large multinucleated cells, and species that pass through multicellular stages. Their hallmark features is the use of pseudopodia, temporarily extensions of the cytoplasm that can form anywhere on the cell surface. These structures anchor to a surface, and the cell then flows to cytoplasm into its extension, pulling the entire cell forward. This movement relies on cytoplasmic streamlining, a process also used inside cells to distribute nutrients and organelles. Gymnoamoeba, also called amoebas, include both naked forms and amoeba protected by shells known as tests. The familiar amoeba Proteus is a classic example of a naked amoeba and is already quite large compared to many other protists. Even so, it is dwarfed by members of the genus Paleomyxa, which can be many times larger and contain hundreds of nuclei within the cell. Remarkably, Paleomyxa has lost its mitochondria entirely and insists relies on bacterial endosymbiosis for metabolic functions. This kind of mitochondrial reduction or replacement is not unique to Gymnoamoeba and highlights how flexible eukaryotic cell evolution can be. Slime molds belong to Amoebozoa and are famous for looking and behaving a bit like fungi, even though they are not closely related. These similarities arose through convergent evolution, meaning similar environmental pressures led to similar solutions. One key similarity is the ability to form spore producing fruiting bodies when conditions become stressful. Slime molds are grouped into two main types based on how their cell cycles are organized: plasmodial slime molds and cellular slime molds. Plasmodial slime mold exists as a single giant cell containing many nuclei. During feeding, this plasmodium creeps along surfaces and engulfs food particles. When conditions worsen, it forms sporangia and produces haploid spores by meiosis. Cellular slime molds behave very differently. They have a separate amoeboid cell until food runs out, at which point thousands of cells aggregate into a slug like structure. Some cells sacrifice themselves to form a stalk, allowing others to disperse as spores. Ophisticonts are named for their defining feature, a single posterior flagellum that pushes the cell forward. This is the opposite of what we see in many other protists, whose anterior flagella pull the cell through the environment. Among the protists, Ophisticonts coenoflagellates are especially important because they closely resemble the cells found in sponges and are thought to resemble the common ancestor of the animal. They can live as single celled or as colonies, and they feed using a collar or microvilli that traps bacteria. This feeding strategy appears in sponge collar cells, suggesting a deep evolutionary connection between choanoflagellates and animals. Another protist lineage with the Ophisticonta is the Mesomyocystozoa. These organisms are mostly parasites of fish, though at least one species can infect humans. Their life cycle remains poorly understood, but they are evolutionarily important because genetic data places them very close to animals. In the past, they formed groups of fungi or other protists based on appearance alone, which shows how misleading morphology can be. Ophisticonta are the last supergroup with what we consider a typical eukaryotic cell structure. Rhizaria are a supergroup of protists best known for the distinctive pseudopodia. Instead of broad-lobed extensions like those seen in Amoebozoa, rhizarians produce thin, thread-like or needle-like pseudopodia that radiate from the cell. Many rhizarians also build elaborate protective shells called tests made from materials such as calcium carbonate or silica. These structures are not just beautiful as they are ecologically important. When rhizarians die, their tests sink to the ocean deep waters, effectively locking carbon away from the atmosphere through a process known as biological carbon pumps. Some rhizarians, especially foraminiferans, are also unique among eukaryotes in participating in the nitrogen cycle through denitrification. Foraminiferans, commonly called forams, are single-celled heterotrophic protists that can range from microscopic to seemingly large, sometimes even visible to the naked eye. Many resemble tiny snails because of their protective shells, known as tests. These tests are porous and usually reinforced with calcium carbonate. Through the pores, forams extend their pseudopodia that allow them to move, capture food, and gather materials to build their shells. Some forams even house photosynthetic algae inside their tests, gaining nutrients from them. Radiolarians are another striking group within the rhizarians. Best known for their beautiful glass like skeletons made of silica, these shells often display radial or bilateral symmetry and are among the most intricate biological structures produced by a single celled organism. Radiolarians extend thin needles like pseudopodia that radiate outwards and reinforced by microtubules, allowing them to trap food particles in the surrounding water. When radiolarians die, their silica shells sink to the ocean floor where they can accumulate in extremely thick sediments layering over geological time. Small remnant of the engulfed green algae structures trapped between membrane layers. At the opposite end of the spectrum are the vampirolites, often called vampire amoeba, which feed on puncturing other cells by pseudopodia and extending into their contents. Chromialveola is a proposed supergroup based largely on genetic evidence rather than obvious shared morphology. The idea is that all chromialveolates descended from a common ancestor that engulfed a photosynthetic red alga, making this a case for secondary endosymbiosis. Because of this, chromialveolates originally had red algae derived plastids. However, some members have since lost those plastids or even the gene associated with them, which make this group less certain. For that reason, chromialveola is best thought of as a working hypothesis rather than a fixed clarification. Alveolates are one of the best supported supergroups of eukaryotes based on molecular data. What unites them is the presence of alveoli, flattened membrane-bound sacs located just beneath the plasma membrane. While their exact function is still debated, alveoli play roles in maintaining cell shape and regulate water balance. Within this group are three very well-known protist lineages: dinoflagellates, apaxicomplexans, and ciliates. Despite their very different lifestyle, these organisms share deep cellular similarities. Dinoflagellates are among the most diverse and ecologically important protists in aquatic systems. Some photosynthesize, others consume prey, and many can switch between strategies. Many species are encased in cellulose plates that act like armor. Their two flagella sit in perpendicular grooves, producing a distinctive spinning motion as they move. Dinoflagellates are a major component of plankton and form the basis of many aquatic food webs. Dinoflagellates are also famous for their unusual cell biology. Their nucleus, called a dinokaryon, contains permanently condensed chromosomes that lack the typical histone protein seen in eukaryotes. During division, the nuclear envelope stays intact, which is highly unusual. Ecologically, dinoflagellates can have dramatic effects. Some species produce bioluminescence, causing waves to glow blue at night. Others form massive algal blooms known as red tides, which can release toxins that kill fish, birds, and marine mammals and pose serious risks to human health and fisheries. The apaxicomplexans and ciliates represent two very different alveolate lifestyles. Apaxicomplexans are entirely parasitic and use specialized structures called apical complexes to invade host cells. They retain a modified plastid, an epicomplex, which is essential for survival. Plasmodium, the malaria parasite, is best well known example. Ciliates, by contrast, are free living and highly complex single cells. Their cilia allow precise movement and feeding, and spores like paramecium have specialized structures for digestion and osmoregulation. Paramecium is famous for something called nuclear dualism, meaning that each cell contains two different nuclei and completely different roles. The micronucleus functions as a genetic archive and is essential for sexual reproduction, but it does not actively express genes. All gene expression happens in the macronucleus, which controls metabolism, growth, and asexual reproduction. The macronucleus is multiploid and divides amitotically, meaning chromosomes are not evenly distributed during division. Because of this, the macronucleus gradually becomes genetically unbalanced and must eventually be replaced through sexual reproduction. Sexual reproduction in paramecium occurs through conjugation, a highly structured process focused on renewing the genetic system. Two mating types connect and exchange genetic materials through a cytoplasmic bridge, Inside each cell, the macronucleus undergoes meiosis, producing four haploid nuclei, three of which are discharged. The remaining nucleus divides. One haploid nucleus is exchanged between the cells. Fusion of these nuclei creates a new diploid micronucleus. The old micronucleus breaks down, and the new one is then built through multiple divisions and extensive DNA editing. After two cells, refresh paramecia, restoring the nuclear balance and genetic integrity. Stromatophiles are a major subgroup within the chromoalveolates and include a wide range of organisms from microscopic unicellular forms to enormous multicellular seaweed. What unites this group is a distinctive type of flagellum that has hair-like projections, which gives the group its name. Many stromatophiles also possess a second smooth flagella. Their chloroplasts originate from red alga through secondary endosymbiosis, linking them evolutionary to other photosynthetic chromoalveolates. Diatoms are among the most important photosynthetic organisms in the aquatic ecosystems. Each diatom is enclosed in a beautiful pattern shell made of silica, which preserves well in sediments. When nutrients are abundant, diatom blooms in massive numbers, and when they die, the silica shell sinks, carrying carbon with them to the ocean floor. This makes diatoms key players in regulating atmospheric carbon dioxide. Green algae are similar in being largely unicellular and planktonic, but they owe their golden color to carotenoid pigments. Brown algae represents the most structurally complex members of the stromatophiles. These organisms are fully multicellular and dominate many marine coastlines and seaweeds and kelp forests. Some species have evolved structurally and resemble plant roots, stems, and leaves, although they are not true plant tissues. Giant kelps are among the fastest growing organisms on Earth, with stipes that can reach tens of meters in length. Like green algae in plants, brown algae shows alternation of generations with distinctive haploid and diploid stages that many differ dramatically in appearance. Excavata is a supergroup of mostly single-celled protists that often show an asymmetrical shape. Many species are named for their presence of a feeding groove that appear excavated from one side of the cell and is used to capture food particles. This group includes a wide range of lifestyles, from free-living predators to photosynthetic organisms and important parasites. Excavata is divided into diplomonads, parabacillids, and euglenozoans. One of the defining themes of this group is organelle modification. Many members have highly reduced or altered mitochondria, while photosynthetic excavates acquire chloroplasts from green algae through secondary endosymbiosis. Diplomonads are a group of excavates best known because they include Giardia lambia, which is a common intestinal parasite. For a long time, diplomonads were thought to lack mitochondria entirely. We now know they contain mitosomes, which are highly reduced mitochondrial remnants. These organelles no longer function in respiration, but instead play roles in iron and sulfide metabolism. Diplomonads live in anaerobic environments and rely on pathways like glycolysis to generate energy. Structurally, they are unusual in having two haploid nuclei per cell and multiple pairs of flagella, which help with movement and attachment to the host intestines. Parabacillids are another excavate lineage adapted to anaerobic environments. They are named for their parabasal apparatus, a structure that links the Golgi complex to the cytoskeleton. These organisms also have axostyles, a supportive rod that can extend through the cell. Like diplomonads, parabacillids have modified mitochondria, but in this case, the organelles are hydrogenosomes, which generates ATP anaerobically and release hydrogen gas. Trichomonas vaginalis is the most well-known parabacillids and cause trichomoniasis in humans. Other parabacillids play beneficial roles in nature, such as digesting cellulose in the guts of termites and remnant animals. Euglenozoans are a very diverse group of excavates that includes organisms with a wide range of nutritional strategies. Euglenoids like Euglena are especially well known because they can switch between photosynthesis and heterotrophy depending on light availability. They use flagella to swim towards light detected by an eye spot. When light is present, their chloroplasts carry out photosynthesis, but in dark, those chloroplasts shrink and shut down, and the cell absorbs organic nutrients instead. Euglena has also a distinctive pellicle made up of protein bands attached to the cytoskeleton, which gives the cell strength and sacrificing flexibility. Another major lineage within the Euglenozoa is the kinetoplastids, which include several medically important parasites. These organisms are named for a kinetoplast, a highly modified mitochondrial plaque with multiple circular DNA molecules. Trypanosoma brucei is the most well known example and causes African sleeping sickness. The parasite is transmitted by the tsetse fly and has a complex life cycle and alternates between humans and insects. Once inside the human host, it multiplies in the blood, lymph, and spinal fluid, eventually affecting the nervous system. Without treatment, the disease progresses to coma and can be fatal, making kinestoplastitis a major concern in human health. Protists are the unsung heroes of the aquatic ecosystem. As plankton, they form the base of nearly all marine and freshwater food webs, feeding mostly aquatic species either directly or indirectly. Many are photosynthetic and act as primary producers, converting light energy into chemical energy that supports entire ecosystems. In fact, one quarter of all photosynthesis on Earth is carried out by protists, especially dinoflagellates, diatoms, and multicellular algae. Without protists, aquatic food chains would collapse and global energy flow and carbon cycling would fundamentally be altered. Photosynthetic protists are critical producers of food and many other organisms. A classic and ecologically important example involves zooxanthellae, which are photosynthetic dinoflagellates that live inside coral polyps. These protists use sunlight to fix inorganic carbon through photosynthesis and share the resulting nutrients with the coral hosts. This energy allows corals to grow, reproduce, and build massive calcium carbonate reefs, even in nutrient poor tropical waters. In return, the coral provides protection and access to light, making it tightly linked and highly efficient symbiotic relationship. Saprobiotic protists specialize in feeding on non-living organic material such as dead organisms and their wastes. By breaking down this material, they release inorganic nutrients back into the soil and water. Those nutrients can be reused by plants and other producers. This recycling process is essential for maintaining ecosystem functions and supporting new growth. Without saprobes, including protists, fungi, and bacteria, organic carbon would remain trapped in dead matter. Nutrients would become unavailable and food chains would eventually collapse. Protist parasites are not just a concern for human health, they also have enormous agricultural consequences. A classic historical example is the Oomycetes Plaspora viticola, which infects grape plants and causes downy mildew. In the 19th century, this disease spread rapidly through Europe vineyards and nearly collapsed the French wine industry. The outbreak showed how vulnerable food systems can be to protist pathogens, especially under favorable environment conditions like cool, wet climates. These unappetizing remains are the result of infections by Phytophthora infestans, an Oomycetes responsible for potato late blight. This pathogen attacks both the leaves and the tubers of potato plants, causing rapid decay that makes the crop unusable. The disease spreads especially fast in cool, moist environments, which plays outbreaks to devastate entire fields. Historically, potato blight is infamous for its role in the Irish potato famine, but it remains a serious agricultural problem today. Start by thinking about where protists fit in global energy flow and nutrient cycles. Many protists are photosynthetic, so ask yourself what that means for carbon dioxide levels. Then think about oceans as a major carbon reservoir and how microscopic organisms have carbon from the surface of deep waters. Consider not just living protists, but what happens after they die. I consider these processes to be long term regulation of Earth's atmosphere and temperature.